Welcome back to Pill Talk. In today's presentation, we will discuss about genetic information and protein synthesis. So, genetic information. Genetic information is stored in the DNA as sequence of nucleotides. So, a gene is a segment of DNA that specifies the amino acid sequence of a protein. So, all the genetic material of an organism is collectively known as its genome. Chromosome houses DNA and specialized proteins called histones. So these histones helps organize and compact DNA into structures known as nucleosomes, allowing it to fit inside the nucleus. So protein synthesis. So protein synthesis is the process of converting genetic instructions into functional proteins. So it involves two key steps. One is transcription and other one is translation. Transcription is the step where DNA is copied into RNA, and translation is the step where RNA directs the assembling of proteins. So now let's talk about the steps involved in transcription. So in transcription, RNA polymerase binds to a promoter region on the DNA. So it separates the strands and uses one as a template to form a complementary RNA strand. So this RNA, after splicing, becomes messenger RNA, and it is ready to carry genetic instructions to the ribosome. You can take a screen snip of that slide so you can read afterwards. Next is the translation. Translation occurs in the cytoplasm, so the messenger RNA binds to a ribosome where transfer RNAs deliver amino acids. So these amino acids are joined into a growing polypeptide chain. It will follow the sequence dictated by the messenger RNA. These are total 9 steps involved. So messenger RNA moves to the ribosome and it follows to the last step, post-translational modification. You can take a screen snap of that slide so that you can read afterwards. So as you can see, after synthesis, protein fold into their functional shapes with the help of chaperone proteins. Some also undergo post-translational modifications such as chemical additions to become fully active. Let's talk about protein degradation. So proteins have varied lifespans. Damaged or surplus proteins are tagged with ubiquitin and directed to proteasomes, where they are broken down into smaller peptides for recycling. And now protein secretion. Proteins synthesized in the cell can be secreted after being processed in the endoplasmic reticulum and Golgi apparatus, and they are packaged and transported to their final destinations through these key stages. Number 1 is protein synthesis. Number 2, post-translational modifications in the endoplasmic reticulum, transport to the Golgi apparatus, sorting and packaging in the Golgi, and number 5 is exocytosis. And if we talk about some examples of a biological importance of protein secretion, number 1 is hormone and enzyme secretion. So proteins like insulin, which is also our hormone, it is also secreted in this way, and digestive enzymes are secreted in this way. And number 2 is immune response. Immune response involve immune cells that secrete antibodies and other proteins, other cytokines, other cells, lymphocytes, etc. They also secrete in this way, and other example can include as cell communication. Cells secrete signaling proteins that bind to receptors on the other cells. It facilitates processes like growth, repair, and differentiation. Next is DNA replication. So DNA replication prepares a cell for division. Helicase unwinds the DNA, and DNA polymerase synthesizes new strands complementary to the original. So this process ensures that each daughter cell inherits an accurate copy of DNA. So the first step in DNA replication is initiation. Initiation process involves an enzyme called helicase, and this helicase prepares, or it can generate a template strand which creates a replication fork. Step number 2 is primer binding. Since DNA polymerase, the enzyme that builds the new DNA strands, can only add nucleotides to an existing strand, so a short RNA primer is laid down by primaries to give DNA polymerase a starting point. Step number 3 is elongation. So DNA polymerase add nucleotides one by one to the growing strand, complementary to the template strand. So DNA replication occurs in 2 directions because DNA strands are anti-parallel. There is one leading strand and number 2 is lagging strand. Leading strand is the one that runs from 5 to 3 direction, and lagging strand is the one that runs from 3 to 5 direction. So on that strand, leading strand, synthesis occurs continuously in 5 to 3 directions. But on lagging strand, synthesis discontinuously occurs in short fragments called Okazaki fragments, since it runs in the opposite 3 to 5 direction. So number 4 step is the joining of Okazaki fragments. So another enzyme called DNA ligase, it will join the Okazaki fragments on the lagging strands. It will seal any nicks in the DNA backbone. So number 5th step is the last step, termination. The replication process concludes once the entire DNA molecule has been copied. Any RNA primers are replaced by the DNA, and enzyme corrects errors in the newly synthesized DNA strand. So let's talk about cancer as it is included in our syllabus outline. Genetic mutations can lead to a cancer. So it is a condition where cells grow uncontrollably. So it can affect epithelial cells, connective tissue, white blood cells. It can lead to carcinoma, sarcoma, or leukemia respectively. And with this, thank you for joining this journey to the chemical composition of the body.